Hello, everyone. Welcome to our brave new world here. This unit will be open for 48 hours. So you'll have until the Wednesday, March 18th at midnight to complete it. We'll have a mix of sort of lecture where I write with you, just like before, and activities interspersed. All right. So I wanted to do a quick kind of check-in about the course now that we've moved online. So let's start there. All right, here's our course, and I just kind of want to remind you of what we've accomplished. We are here. Let me write. Here we go. We are here. And lucky for us, I really think that we've done the two hardest units already, and we were able to do them face to face. Migration is pretty straightforward and shouldn't be too challenging, but of course there will be the discussion board open for you to post questions, and I am happy to make appointments to meet folks by Zoom if you have individual questions that you think would be easier sort of face-to-face, virtually face-to-face anyway. Sexual selection can be a little bit complicated, but it's interesting, so I think it'll help you stay focused and keep with the unit, and then hopefully we'll be back face-to-face by Unit 8. All right, so it's really these two units that we're worried about right now. But I think we're going to be fine. Okay, so let's start as we usually do with a knowledge check. All right, this knowledge check is a bit of a review of genetic drift, so a little practice for your exam. There should be a play pause it window that pops up where you can enter your answer. So I'll give you a few minutes to do that. You won't be able to progress until you've answered in play pause it. All right, now that we're done with our review, let's talk about our new unit. So we're going to focus on the next mechanism of evolution, which is migration. And the unit is going to be divided into two modules, and the split will be about here. The first module will be today, and it's how species are arranged on a landscape. What is migration? What do we mean by it in this class? What are some of the characteristics of it in terms of evolution, and how do we measure its impact? Then the next day, we're going to talk about layering it with natural selection and drift. How does it interact with those two mechanisms? And how do we identify when natural selection is leading to local adaptations? And we can use migration to help us do that. And then we'll talk about migration in the context of conservation, which is one of the applications of it sort of in the real world. So our prior work with evolution has really focused just on evolution sort of in the abstract and not in space, not in the real world, right, where it's on a landscape. And so we know that when you put things on a landscape, and you've probably heard this in your ecology class, of course, that spatial and temporal differences become important. And so the same thing is happening here. Species on a landscape, it's not as easy as it might have seemed for them. Let me get this pen going here. Okay, so individuals do not freely move all over the landscape. Okay, their movement is limited by many things, but here are some examples. So their movement is limited barriers, which can be things like the ocean if you're not, aquatic. Mountains, cities, man-made landscapes are not necessarily appropriate for a lot of critters. Their movement is also limited by their dispersal capabilities. Sorry about the cats in the background. They can also be limited by difference in the environments that aren't barriers but do impact their survival. So environmental differences that impact survival. For example, an iguana, there's nothing keeping an iguana from moving north in Florida, right? But the chance of it surviving the winter, there's not a barrier, but the chance of it surviving the winter in that habitat is very low, right? So that's the difference between a barrier. They can do it, right? They can travel that distance, but there are consequences on their survival for doing that. And so you don't see them much further north, okay? So because of these things, because of limited movement, species are often made up of multiple discrete populations. And these populations are held together by migration. So that are held together by occasional migrants moving between them. And we'll talk about what we mean by held together. In this class, when we use the word migration, we mean something maybe a little different than the general meaning of the word. So migration, for evolution, the migration that matters is movement of individuals and, most importantly, gametes. Right? So if gametes aren't moving, then it's not migration that evolution cares about. So this is distinct from seasonal movement of individuals if you disperse but then come back to the same place every year to have your offspring, then your gametes aren't changing location, right? They're still part of the same population. So what we're really focused on is where individuals leave their offspring. Are their offspring part of a different population than the parent originally was? All right, so a better word for what we mean by migration is really gene flow. And I'll switch between the two of these during this unit. But we mean gene flow when we talk about migration. So gene flow is the mixing of alleles from different populations. And this is the evolutionarily significant type of migration. To get a sense of what migration can do, both within a population and between populations. I want you to read through this scenario and answer the series of questions that pop up on Play Pause It. These questions will also be a little bit of an exam review as you're comparing migration to what drift does in and between populations. So hopefully what you saw through those two questions is that migration is kind of the opposite of drift. So if we're talking about the evolutionary consequences of gene flow, there's two big ones, right? And again, they're the opposite of what drift does, if that helps you organize it in your head. First of all, gene flow prevents populations from diverging. It basically, oops, I just lost my pen again. It basically homogenizes allele frequencies. Across populations. So it makes them more similar to each other. And it erodes genetic differences established by drift and natural selection. And we're going to talk about why there might be population differences by natural selection a little bit later in this unit. All right, so the second big pattern of migration and why it's important for evolution is that it can also increase diversity in a population. So increase by, and this happens by introducing alleles into populations where they didn't exist before. Okay, so basically you're bringing in alleles from other populations. So those slides get us through these first 3 points, and now we're going to focus in on how do we measure gene flow in a population. So environments can take many different forms, but they can be categorized into kind of 2 ways. They can be discrete or they can be continuous. Discrete populations have clear boundaries. Discrete environments. Where continuous environments kind of just bleed into each other. So it's hard to tell where one ends and the other begins. So we can calculate migration for both of these, but it's easiest, and what we will focus on is calculating migration for discrete environments. Okay, so in discrete environments, the migration rate which we're going to call M, is just the fraction of individuals in a population no S, that arrive from another population each generation. All right, so it's all the new individuals that come into the population each generation. And we can use that migration rate to figure out evolutionary impact, all right? So that's really what we're focused on here. So we're trying to figure out the difference. So the change, triangle means change. Change in, say, the P allele due to migration. And there's an equation for that. So it's this change is equal to M times the frequency of P in migrants minus the frequency of P in a focal population. And this is before the migrants arrived. All right, so it's the migration rate, so the number of individuals that are coming in, times the frequency of P in the migrant population, minus the frequency of P in the population that they're arriving to. Okay, so let's look at a real world scenario to help us understand what this equation is telling us. So we're going to focus in on water snakes in Lake Erie. Lake Erie is a great lake in the Midwest. And in this Great Lake, water snakes have colonized a bunch of different islands. They're also on the mainland. And so every year, snakes from the mainland come out to these islands. So they migrate out to the islands. On the mainland, the snakes look different than on the islands. So on the mainland, the snakes are banded. So you can see in this picture, some of the snakes have dark bands on them. But on the island, they are mostly or exclusively unbanded. So the snakes there that don't have any markings on their back are island snakes. The banding pattern is thought to be controlled by one gene that we're going to call, and it'll have a P and Q allele. And it's the P allele that creates the bands. So to understand how the island population will evolve in appearance due to migration, researchers collected the data they need to complete that equation, right? So they collected for each of the islands, which was South Bass, Pele, North Bass, and the Kellys. They collected the migration rate, so what percent of the population is migrants from the mainland each year. They calculated the frequency of the P allele in those island populations, but they also got it for the mainland, right? So the mainland was 0.7. And then from that, we can calculate the change in P, so how much impact migration is going to have in this population. So I'm going to do the first one, and then you're going to do the second one, okay? So let me show you how to do this. So I'm going to do South Bass. So the migration rate is 25%, so that'll be 0.25 times the difference between the mainland frequency and the frequency of the allele in the current population. So the mainland's P allele frequency is 0.7, and then my island is 0.3, so it's 0.7 minus 0.3, which equals 0.1. So 0.1 is the change in the P allele due to migration. All right, so we can write that in our table. And now you're going to do the next island on your own. All right, great work. I'm now giving you the entire table with all the outcome, the changes in P. And you're going to do an activity where you try to list the factors that are impacting the strength of migration, the impact of migration on evolution. So when is it strongest and when is it weakest? Those sorts of things. So through that activity, I hope that you saw at least one of these two patterns, all right? So the change in the frequency of P due to migration is influenced by migration rate, and by how different migrant allele frequencies are from focal population frequencies. Okay, so migration is going to have a greater evolutionary impact when there are higher migration rates. And what this is going to do is more quickly erode genetic differences between populations. And then how different migrant allele frequencies are from the focal population is the other main factor that impacts migration's evolutionary potential. And basically here, the greater the differences between migrants and focal pop, the more change in allele frequencies migration will cause. Now this all assumes that the migrants actually successfully mate. So let's make that clear. We're assuming migrants successfully mate. Right? Because again, we're interested in where they're leaving their offspring. So if they don't successfully mate with anyone from the original population, they have no evolutionary impact. Okay, so that's it for this unit. We're gonna pick up on Thursday, the second half, which will be a little bit longer once we've all kind of gotten the hang of this. Thanks for your patience with me while I was kind of figuring this out on the fly. And I look forward to seeing some of you during office hours, and otherwise, I'll be available to you on Thursday. Talk to you all later. Bye.